All right, hello everybody. This is a recording for Biology 105. This is a quick follow-up video in fall of 25 for lecture 32. I'm recording this on November 14th, right before our exam next Monday. I just wanted to emphasize a couple of things that we talked about in lecture, but we didn't really have time to cover. I'll keep it quick. We're going to just do this real fast, and I hope it's helpful to you. Okay. So we ended up getting through almost all of the topics on the study guide, including blood types and codominance and pleiotropy and epistasis, defining all of those, but we did not really have time to discuss the sex chromosomes and inheritance on the sex chromosomes. So we're going to talk about that. We talked about aneuploidy. I'm just confirming that everything here is good. Okay, so I'm just going to cover those last concepts, focusing on sex chromosomes, which usually you're talking about the X chromosome when you're talking about sex chromosomes. Okay, so there's an X chromosome, there's a Y chromosome, and hey, the X chromosome is big, the Y chromosome is little. So there are a lot of genes that are only present on the X chromosome. So that means that people with two X chromosomes have two copies of those genes. And people with an X chromosome and a Y chromosome have only one copy of those genes. All right? That means that inheritance patterns are going to be different between males and females when when you're looking at characteristics encoded on the X and the Y chromosome. Okay? So the important thing here is going to be looking at the notation so we can make predictions of these inheritance patterns. Okay? So Thomas Morgan had his fly experiments, his super famous foundational work, where he was tracking different phenotypes in flies. So it's kind of like Mendel's work. He took these red-eyed flies and these true breeding white-eyed flies, and he crossed them, and he found that, hey, they've got a 3-to-1 ratio in the F2 generation of red eyes to white eyes. Great. Follows the rules of Mendelian inheritance. Red is dominant to white in eye color. Okay? There's a lot of foundational work. You can check these slides out. People were wondering, like, hey, what are Mendel's dang factors? You know, what's the thing that's actually being inherited here? And at this point, people could see chromosomes. So they were showing, like, oh, well, if we track the behavior of these chromosomes during inheritance and we look at the cells, we can see chromosomes. Anyway, some work gets done. We've got Boveri and Sutton. They're next to their pictures. And they found, well, hey, if chromosome number is different, all right, then there's differences in development of flies. So inheritance must be occurring through the chromosomes. Okay. Thomas Morgan ended up supporting Mendelian inheritance. Well, supported that chromosomal inheritance was occurring. Whatever. Here's our point. Here's what we're going to focus. Morgan does this experiment. Check it out. P generation, true breeding red-eyed, true breeding white-eyed. Okay? Breed them, you get the red-eyed flies. Okay? All of these individuals, I'm going to annotate this slide. Pen. If we're looking at this as classic Mendelian inheritance, sorry, I'm getting a little trouble with my pen here. We would expect these individuals would be dominant, homozygous dominant for the red eyes, and these individuals here will be homozygous recessive for the red eyes. Okay? So they've got white eyes. This F1 generation, therefore, should be all heterozygous. And then these offspring here, you should have a 1 to 2 to 1 ratio. 1 homozygous dominant to every 2 heterozygous to every 1 homozygous recessive. All right? These are the predictions. And that did hold, and that did work if you looked at the population regardless of male versus female flies. All right? So if you remember here, For our purposes, XX is going to be the females, and XY is going to be the males. I'm using a weird tablet, so forgive my sloppy annotations. Now here's the thing. The eye color gene is on the X chromosome. So male flies, they only have one copy of that gene. Okay? Female flies have two copies of that gene. Alright? So we're going to look at that in more detail as we keep moving forward. So check this out. You've got a homozygous white-eyed female right here. Let me switch over to a laser pointer. Homozygous white-eyed female here. Okay? Two copies of the gene, both of them for white eyes. Cross this one with a red-eyed male. Now this is what we would call a hemizygous individual. Males are hemizygous for characteristics encoded on the X chromosome because they only have one X chromosome. So this red-eyed male only has one copy of the gene, and it is the red-eyed version. So now we can do a Punnett square here. Okay? You make the Punnett square. Now, the individual, the parent that determines whether you are XX or XY, is the paternal side, the father's side, because you will always receive an X chromosome in the egg cell. The sperm cell can either have an X chromosome or a Y chromosome. You've got a 50-50 chance, right? If you look right here, it's a 50-50, a probability of 0.5 to receive the X chromosome in the sperm, 0.5 to receive the Y chromosome in the sperm. Now over here, we've got the X chromosomes being inherited in the egg cell. Every egg cell will have an X chromosome, okay? So check this out. All of the male offspring will be inheriting the X chromosome from their maternal side. Because if they ended up being male offspring, they must have received the Y chromosome from the paternal side. Okay? The female offspring are going to inherit an X chromosome from each parent. So it follows different rules here. What you end up seeing are phenotypic ratios differ between males and females. Okay? Oops. I've just moved the entire screen instead of my face cam. I'll put myself right up here. There we go. So, what Morgan figured out was that eye color was on the chromosome that determined sex. So we've got males, all right? They get the eye color gene only from the mother because the egg cell always has an X chromosome. If the sperm cell has an X chromosome, you have female offspring. You have a daughter. All right? We good so far, I hope? That the males are what we call hemizygous, meaning they only have one copy of the allele. Okay, great. There's a lot of additional stuff here. I encourage you to look at the PowerPoint. If this didn't make sense, double back, pause, look at these slides, right? They've got a lot of different notes on them. So this is called sex-linked or X-linked inheritance, these different rules. And you get different predictions when you're talking about crossing. You get different predictions and you analyze this information differently. So there are some disorders that are on the X chromosome. We call them X-linked disorders, like color blindness and hemophilia. Here we've got an example of a big royal family with Tsar Nicholas II of Russia here. Check this out, right? We had all of these different couplings, okay, between people who did not have hemophilia. Here's the thing, right? If any of these men had the gene for hemophilia, it would be expressed. This is an X-linked recessive disorder. Therefore, you need two copies of it. Okay, you need to be all recessive in order to express the recessive phenotype, in order to have the disorder. But only females have 2 copies. Males only have 1 copy of this gene. So that means that if you're a male and you inherited the recessive allele, you express the recessive phenotype because you only have 1 copy of each X-linked chromosome. Okay? So check this out. We've got all of the Queen Victoria, Alice, Alexandra. They're all heterozygous for hemophilia. They've got a dominant allele, they've got a recessive allele. That means that they're passing on, one or the other. Now, if you, the male in the coupling event, okay, always has the dominant version of the allele because they don't have hemophilia. So these individuals, I'm going to actually just annotate this slide real quick with a little pen. Let's look at their genotypes. So here's Albert. He does not have hemophilia. I'm just going to use the letter N. I don't know why I chose N, but we're just going to go with it. Queen Victoria, she did not have hemophilia either, but she's actually a carrier. So she's got 1 copy of the recessive allele on her other X chromosome. They have a daughter who must receive the dominant allele from the paternal side in the sperm cell, but received the recessive allele from the maternal side. And there was a 50% chance that any daughter that these two had would be a carrier for hemophilia. Okay? Because this is a 100% chance in a daughter that she received the dominant allele from the paternal side, but there's a 50-50 chance that she would receive the recessive allele from the maternal side. Okay? So this is the phenotype of the daughter, heterozygous. Alice couples with Louis or Lewis. Lewis does not have hemophilia, therefore he must have this genotype, the dominant allele on the X chromosome. The dominant allele on the X chromosome, and then no allele on the Y chromosome, hemizygous. They have a coupling event, and they have Alexandra, who also is heterozygous, okay, 1 dominant, 1 recessive, who then couples with Tsar Nicholas II of Russia, and then they have a son, okay? Now, their son Alexis has hemophilia, and the Tsar says, well, nobody in my family's had hemophilia, this is your fault, what's going on? And then she says, well, nobody in my history has had hemophilia, so I don't know what's going on, And let's take a look, let's draw a little Punnett square, and see what the likelihood of this was. So we've got the egg cell on the top. Here's the Punnett square. And I know I've told you in lecture multiple times, like you don't need Punnett squares. We're just going to visualize this in this way. Okay? Oops. I did that incorrectly. That should be a Y. So here's the sperm cell over here. Oh my gosh. Here's a sperm cell. And over here we've got the big old egg cell. Okay, on the top. So if the individual is XY, these are, right here, the potential sons. These are the male offspring. So what was the likelihood that when they had a son, that son would have hemophilia? Well, 50-50, right? Because half of the time they'll receive a dominant allele in the egg, half of the time they'll receive a recessive allele in the egg. If they had a daughter, what's the likelihood that she would have hemophilia? It's actually 0. There's a 50-50 chance that she would be a carrier, but there's no way for her to have hemophilia, based on the Punnett square we've got right there. Okay? I'm just going to keep moving through these slides. Okay. So here's, that's why I went with N, because it's on the next slide. Okay. So this is the notation we use for talking about X-linked characteristics, is we've got the X's, right? And then if the X has a capital letter, it's dominant, the dominant. If the X has a lowercase letter, it's the recessive version of the allele. And then Y has no allele, all right? Doesn't have it. Going to keep moving forward. Dosage compensation. Okay, so people like this tortoiseshell cats. All right, tortoiseshell cats or calico cats. Right? People say, oh, they're all female, and why is that? Well, because of X chromosome inactivation. Okay, so oftentimes, think about this, right? In individual, you for a lot of your genes, you don't need two copies of every gene on the X chromosome. If that was the case, then me personally, I've got an X chromosome and a Y chromosome. If I needed two X chromosomes to function as a human and exist, then I wouldn't do so well, right? So you have extra set of instructions in a lot of these. So in many of your cells, X chromosome inactivation will occur, where X chromosome stays active and the other X chromosome condenses into this thing called a bar body. Alrighty. So it kind of just clumps up. So check this out. Here's the cat example right here, where early on in the embryo, the cat's got these two X chromosomes. One's orange fur, one allele for black fur. Now, as the organism develops, okay, as the embryo develops, you end up with inactivation of some chromosomes, and it leads to this sort of what we call a mosaic character, where some tissues in the cat have the version of the X chromosome with the black fur allele being expressed, and some cells in the cat have the version of the chromosome with the orange allele being expressed. All right, linkage analysis we're not worried about. We'll talk about that later. Genetic disorders, we talked about aneuploidy. Polyploidy is, multiple copies of each chromosome beyond being diploid, and it happens in plants all the time. We're going to talk about these other things later. There's also one thing I just want to mention it real quickly. That's extranuclear DNA, which we've already talked about when we talked about mitochondria and chloroplasts. One of the pieces of evidence that they used to be free living things is that they had is that they have their own genome. Okay, so there's something called the nuclear genome, and that's the central for you it's your 23 chromosomes. Okay, but then inside of your cells you've got mitochondria. The egg cell is big. Okay, it contributes the bulk of the cytoplasm to the zygote, and therefore the egg cell contributes all of the mitochondria or all of the chloroplasts. So all of your mitochondria were inherited from the egg cell. So if there's a mitochondrial disorder, that comes from the egg cell. All right? It's pretty interesting. I don't intend to have any exam questions about it, but I want it to be brought up. So I know that this is, a little bit... 1 second here. I know it's a little bit rough to be like, well, we're going to have a video recording of this topic that then is going to be on your exam. So I wanted to give you a nice practice question, okay, that's similar to the one on your exam, so you can be prepared. So if you watch the video, you get a little reward. We're going to do a practice problem right now. Okay, let's say that we've got two individuals. Get my annotations out here. All right, ready? Okay. Oh, there's a laser pointer. Pen. Okay, color blindness. All right. X-linked recessive disorder. That means that you must have, there are two possible genotypes for color blindness. There's this one or this one. So the homozygous recessive or the hemizygous. And right here would be the female, and right here would be the male. Both of these genotypes would lead to colorblind offspring. So let's say that two parents without color blindness. All right. Have a son with color blindness. Okay. What are their phenotypes? What are their genotypes? Fair enough question. Let's look at the son. So the son must have this genotype right here. It's the only possible genotype for the son to have to be colorblind. If the offspring is a son, then he must have received the Y chromosome from the sperm cell, from the paternal side. So that means that he received this recessive allele from the maternal chromosome. So this is the paternal one's easy. Paternal, all right, normal, not colorblind, must have this. Normal vision, must have this. Maternal side, all right, the maternal side, normal vision, so she must have one copy of the dominant allele, but she passed on a recessive allele. All right? Now let's take these same parents, maternal and paternal. Okay? They both got normal color vision, but the mother is a carrier for color blindness. What's the likelihood? Okay, we're going to do a little quick problem here. What's the likelihood of a colorblind daughter? Fair question. So, the colorblind daughter. She has a 50% chance of receiving a recessive allele from the maternal side. So that's a probability of 0.5. The probability of receiving a dominant allele from the positive side, from the father's side, is 1. There's only one possible genotype that this daughter could have. That's heterozygous. Oh, actually, no. In order to be colorblind, she would have to be homozygous recessive, right? This is fun. I like making mistakes on a recording. What's the likelihood of a daughter being colorblind? Well, there is a 0.5 probability of receiving the recessive allele in the egg cell times a 0 probability of receiving a recessive allele from the sperm cell. Therefore, the probability is a straight up 0. They can't have a colorblind daughter. All right? That's the sort of problem that I would ask you on the exam that has to do with X-linked inheritance. Okay. That went a little longer than I was hoping. What did we end up with? We ended up with like 23 minutes. Okay, well, I hope it wasn't too bad to sit through. I hope you got something out of it. And I wish you the best of luck on next week's exam. Keep working on that study guide, and thank you all for watching this video. Bye-bye.